हेलो स्टूडेंट्स, वेलकम टू बिरेन्स एकेडमी। एग्जाम तो बहुत पाख आसीगला आम किछिटा क्वेश्चन आंसर डिस्कस करदे। सो चैप्टर आज नहीं मु पेपर 2 यूनिट 1 सेल उपरे किछि आउ स्पेसिफिक टॉपिक नहीं सेल अच्छी आमर जोटा स्ट्रक्चर एंड फंक्शन ऑफ प्लाज्मा मेम्ब्रेन। सेकंड जिनस रहा आमर सेल साइकिल एंड सेल डिवीजन। एतिकी ऊपर ही क्वेश्चन किसी नहीं जोटा कि क्वेश्चन डिस्कस कर दूची। सो फर्स्ट जिनस आसला आमर प्लाज्मा मेम्ब्रेन इज मेड अप ऑफ। तो बहुत बेसिक क्वेश्चन आमर ओपीएससी लेवल रे अच्छी। तो एकदम बेसिक क्वेश्चन, एकदम इजी क्वेश्चन नहीं। तो क्वेश्चन गुड़ाक बारंबार रिवाइज करके मन रखिदबा। सो प्लाज्मा मेम्ब्रेन इज मेड अप ऑफ लिपिड प्रोटीन एंड कार्बोहाइड्रेट्स, जोटा कि आम जाचे आम कहूचे फॉस्फोलिपिड बाइलेयर ता सांगरे ग्लाइकोप्रोटीन्स रोचे। सो ग्लाइकोप्रोटीन्स कहले सुगर प्लस प्रोटीन और लिपिड बाइलेयर लिपिड एसिड। सो लिपिड प्रोटीन एंड कार्बोहाइड्रेट्स। द नेक्स्ट नंबर, which is the latest model that is proposed to explain the structure of plasma membrane, that is fluid mosaic model. Who proposed the fluid mosaic model of plasma membrane? Singer and Nicholson. Assertion and reason question. The fluid mosaic model proposed by Singer and Nicholson indicated that quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. Reason is, in the cell wall of a young plant cell, the primary wall is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner side of the cell. So, which of the following is true? That is, both assertion and reason are true. Fluid mosaic model Singer Nicholson propose करथिले, obviously quasi fluid nature सेटा भी होते ठीक। देन आम प्लांट सेल कथा कहूचे, सेल वॉल कथा। सो प्राइमरी वॉल इज कैपेबल ऑफ ग्रोथ, ठीक अच्छी। तो असर्शन भी ठीक, रीजन ठीक, किंतु कहीं क्या ये एक्सप्लेनेशन करूना। सो बोथ असर्शन एंड रीजन आर ट्रू, बट रीजन इज नॉट द करेक्ट एक्सप्लेनेशन ऑफ असर्शन। Which of the following statement is not true for plasma membrane? It is present in both plant and animal cell. एटा ठीक अच्छी। सो भूल नुह। Lipid is present as a bilayer in it. A true sentence. Then proteins are present integrated as well as loosely associated with the lipid bilayer. एटा मध्य true statement. Carbohydrate is never found. ये हला हमरो false statement, जोटा कि हमें कहते is not true. सो कार्बोहाइड्रेट हमें पाइबा, हमें स्टार्टिंग रे हमें कहीदेले प्लाज्मा मेम्ब्रेन रे प्रोटीन, कार्बोहाइड्रेट्स, लिपिड देखा पाइबा। नंबर 6, रीड द फॉलोइंग स्टेटमेंट्स एंड चूज द करेक्ट ऑप्शन। स्टेटमेंट 1, अ सेल मेम्ब्रेन शोज फ्लूइड बिहेवियर। ओके। देन स्टेटमेंट 2, अ मेम्ब्रेन इज अ मोजाइक और कंपोजिट और डाइवर्स लिपिड्स एंड प्रोटीन्स। सो ये हमें कहूचे स्टेटमेंट 1 भी ठीक अच्छी, स्टेटमेंट 2 भी ठीक अच्छी। सो बोथ स्टेटमेंट 1 and 2 आर करेक्ट। ऑप्शन सी। Which of the following controls permeability, that is cell membrane, जोटा कि आम कहूचे ट्रांसपोर्टेशन जोटा रेस्पॉन्सिबल प्लाज्मा मेम्ब्रेन। सो परमिएबिलिटी शो करे, दैट इज आम कहूचे ना कि किछिटा को से सिलेक्ट करिकिना से अलाउ करिवो। सो परमिएबिलिटी को शो करछि प्लाज्मा मेम्ब्रेन। सिलेक्ट द इनकरेक्ट स्टेटमेंट फ्रॉम द फॉलोइंग। In plasma membrane, hydrophilic polar heads of lipids arranged towards the outer side and the hydrophobic tail towards the inner side. हमको ये इनकरेक्ट स्टेटमेंट खोजिबार अच्छी। सो ये जोटा कहला हम ये एकदम करेक्ट स्टेटमेंट, कहीं ना प्लाज्मा मेम्ब्रेन हेड हाइड्रोफिलिक किंतु जो टेल रोचे सेटा हला हाइड्रोफोबिक। देन इन प्लाज्मा मेम्ब्रेन peripheral proteins lie on the surface of the membrane while the integrated proteins are buried in the membrane. Obviously एटा ठीक अच्छी, कारण peripheral माने periphery रे रहिबो, integrated protein माने membrane माने embedded रहिबो। Then आसिला neutral solutes may move across the membrane by the process of simple diffusion against the concentration gradient. Against the concentration gradient लिखा है तो simple diffusion हबो नाही। तो ये रहला हमर incorrect statement. Then polar molecules can pass through the non-polar lipid bilayer and do not require a carbon. एटा मध्य incorrect statement. So option B, जोटा कि हमरा incorrect statement शो करूछ। Ion carriers are located in cellular spaces. Which one of the following is correct for the transmembrane proteins in lipid bilayer? Okay. Transmembrane protein पाई आमे कहबा absent in animal cell. हमको correct statement खोजिबार अछि तो एटा भूल. They act as channel protein. Then they are absent in plant cell. एटा मध्ये भूल. They are only extremely located. एटा मध्ये भूल. So correct statement हैला they act as channel protein. Read the following statements and choose the correct option. Statement 1. Active transport is an energy dependent process. Okay. Then in active transport ATP is utilized. So obviously statement 1 and statement 2 both are correct. Active transport रे ATP दरकार हुए कहीं सेटा ना against the concentration gradient जाउचे एठी ऊपर आमे गोटे देखिथिले स्टेटमेंट लिखिथिला against the concentration gradient तो एटा simple diffusion नुह बोली आमे कहले. So that is active transport. Select the correct statement from the following regarding cell membrane. Na plus K plus ion move across the cell membrane by passive transport. Rule. Active transport से सोडियम पोटासियम एटीपेज जरूरी है सोडियम पोटासियम पंप को ऑपरेट करिवार पाई. So that is active transport. The membrane of erythrocyte has approximately 60 to 70% protein. नहीं भूल अछि. Then lipids are arranged in a bilayer with polar head towards inner side. नहीं भूल. हाइड्रोफोबिक हमें कहूचे inner side को. Fluid mosaic model of cell membrane was proposed by Singer and Nicholson. Correct statement. Algae have cell wall made up of cellulose, galactans, mannans, hemicellulose, pectins, proteins. Boolela. Pectin, cellulose, protein, cellulose, hemicellulose, protein. So cell wall is made up of cellulose, galactans and mannans. In the cell wall of a young plant cell, the primary cell wall is capable of growth. This is the assertion. Then reason plant cell wall is made up of cellulose, hemicellulose, pectins, proteins. तो ये हमें देखिए को पाउचे रीजन भी ठीक रहिला. सेल वॉल ऑफ योर प्राइमरी सेल वॉल इज कैपेबल ऑफ ग्रोथ ठीक अछि किंतु केही कार रीजन नो करेक्ट एक्सप्लेनेशन नो. सो बोथ assertion and reason are true, but reason is not the correct explanation of assertion. Option B. Which of the following parts of a young growing plant cell gradually forms the layer inner adjacent to the middle lamella? Which of the following parts of a young growing plant cell okay तार को पार्ट ता रहउची जे कि गोटे लेयर फॉर्म करिवो मिडल लामेला पाखापाखी से रहला हमरा प्राइमरी वॉल जे कि ग्रोथ पई रिस्पांसिबल. Middle lamella is made up of that is calcium pectate. The filaments associated with cilia and flagella are constituted by that is microtubule. एबे हमरा आसीगला साइटोस्केलेटन ऊपर किछटा क्वेश्चन. cilia, flagella, centriole, centrosome ए ऊपर किछि क्वेश्चन डिस्कस करिवा. Then electron microscopic study of cilium and flagellum revealed that they are covered with plasma membrane and the core structure is called axoneme, possess a number of microtubule running parallel to the long axis. How many number of microtubule arrays are arranged parallelly and centrally? Obviously that is 9 plus 2 microtubule arrangement. The cell organelle involved in the formation of a flagellum or cilium that is mitochondria. माइटोकॉन्ड्रिया जे कि फ्लैजेलम एंड सीलियम फॉर्मेशन पाई ओ सॉरी इधर 9+2 अरेंजमेंट थिला. देन क्वेश्चन कोन आसिला द सेल ऑर्गेनेल इन्वॉल्व्ड इन द फॉर्मेशन ऑफ फ्लैजेलम और सीलियम. ओके फ्लैजेलम और सीलियम पाई के रेस्पोंसिबल आसिला ऑप्शन आसिला माइटोकॉन्ड्रिया, लाइसोसोम, बासल ग्रैन्यूल, गोल्गी कॉम्प्लेक्स. तो माइटोकॉन्ड्रिया नो आंसर हैला बासल ग्रैन्यूल. Then sheath of central microtubule is connected to peripheral microtubules by radial spoke. Then centrioles and centrosomes are present in cells of centrioles and centrosome are present in cells of that is animals. Animal cell रे हमें पाइबा. Then function of centriole is formation of spindle fiber. The function of centrosome is inhibition to cell division, initiation cell division to increase protein synthesis. None of कहीं भी नुहा. So none of these. Centrosome is found in cytoplasm. Match list 1 and list 2 and select the correct answer using the code given below the list. List 1. Microtubules. Microtubules कहा संग्रह में जॉइंट करवा. अच्छा कौन दीजिए करेक्ट आंसर यूजिंग द कोड गिवन बिलो. अच्छा I double I triple I are correct. I and double I are correct. Triple I is false. Okay आमे देखि दबा आईडी. Microtubule देला कौन अच्छी ना structural components of cilia. Okay तो ये ठीक अच्छी. Then centriole store hydrolytic enzymes. नहीं भूल अच्छी. Ribosome store oil and starch in plant. भूल अच्छी. So only option I is correct. Option C. Microtubules are the constituents of centriole, spindle fiber, chromatin. Microtubules are the constituents of centrosome, nucleosome, centrioles. 9. Then cilia, flagella, peroxisome. 9. Then spindle fiber, centriole, cilia. Correct answer. Which of the following is not a function of cytoskeleton in a cell? Cytoskeleton रो function नुह. अच्छा इंट्रासेलुलर ट्रांसपोर्ट देला, मेंटेनेंस ऑफ सेल शेप एंड स्ट्रक्चर देला, सपोर्ट ऑफ ऑर्गेनेल देला, सेल मोटिलिटी देला. साइटोस्केलेटन रो फंक्शन. अच्छा एटा मोस्ट बहुत इंपोर्टेंट क्वेश्चन आसुची. आमर फंक्शन आसिला कौन कौन मेंटेनेंस ऑफ सेल शेप एंड स्ट्रक्चर. साइटोस्केलेटन जी सेल को स्ट्रक्चर प्रोवाइड करे, obviously support the cell organelle, cell motility पाई मध्य responsible. cilia flagella रो काम ही motility किंतु intracellular transport करे नहीं. So this is the option, that is option A, is not a function of cytoskeleton. Then next आसीबा हमें cell cycle ऊपर किछि क्वेश्चन डिस्कस करबा. The sequence of events by which a cell duplicates its genome, synthesizes the other cell constituents and eventually divides into two daughter cells is termed as, that is cell cycle. The duration of cell cycle in yeast is, that is 1.5 hours. ता मने रखिबा 1.5 hours. A cell cycle consists of mitosis, meiosis, prophase, metaphase, anaphase and telophase, G1S, G2 phase, interphase and mitosis. The stage between two successive mitotic division is prophase, telophase, metaphase, interphase, that is interphase. In human cell, the interphase lasts more than -% of the duration of cell cycle, that is 95%. आमे आगरू कही थिले जेहेतु दुईटा फेज कथा आमे कहूचे, गोटे इंटरफेज कहूचे आ गोटे एम फेज कहूचे. तो गोटे हैला इंटरफेज जोटा गोटे प्रिपेरेटरी फेज बोली आमे कहूचे. सो आमे जेतेबेले मु एटा मध्ये पढेला बेले कहिथिली कि आमे जेतेबेले एग्जाम पई प्रिपरेशन करूचे, सो ऑब्वियसली आमे देखो एठी आसीगला हमको ऑलमोस्ट 9 मंथ्स पाखापाखि तुमको मिलीगला टाइम प्रिपेरेशन. किंतु जो एग्जाम ड्यूरेशन दौचे आमे जमती पेपर 1 धरने तुम नेल के 2 आवर्स देल एग्जाम. तो एक्जाक्ट सेमती इंटरफेज जोटा कि प्रिपेरेटरी फेज, तो यहाँ बहुत टाइम निए. आ एम फेज जोटा कि तुम फाइनाल से कौन हुए ना कम टाइम तुमको लगे. सो यहाँ से हिसाब से तुम मन रख लास्ट मोर देन 95% ऑफ द ड्यूरेशन ऑफ सेल साइकिल. देन ये देला सीक्वेंस ऑफ फेज को माने फेज रो करेक्ट सीक्वेंसटा कौन हम. सो करेक्ट सीक्वेंस ये आम कहवा G1S G2M. इन अ माइटोटिक साइकिल द करेक्ट सीक्वेंस ऑफ फेजेस इज G1S G2M. Identify the phase that represents the interval between mitosis and initiation of DNA replication, that is G1 phase. Select the correct statement about G1 phase. G1 phase by cell is metabolically inactive, completely full. Cell metabolically active रहे. DNA in the cell does not replicate. हाँ, it is not a phase of synthesis of macromolecule. Cell stops growing. So DNA replicate हुए नहीं. Identify the correct statement with regard to G1 phase of interphase. G1 phase by reorganization of all cell components takes place. Cell is metabolically active, grows but it does not replicate DNA. ठीक अच्छी. Nuclear division takes place. भूल. DNA synthesis or replication takes place. भूल. Given below is a schematic breakup of the phases or stages of cell cycle. Which one of the following is the correct indication of the stage or phase in the cell cycle? Okay. Correct कोटा अच्छी ना ट्रिपल आई देला कैरियोकाइनेसिस नाई आई भी देला सिंथेटिक फेज एटा एटा तमर आसी जी नॉट G1 देन आसला एटा तमर एस फेज देन आसीबो G2 तो ये ठीक अच्छी करेक्ट इंडिकेशन करुच्ची आई देला साइटोकाइनेसिस नाई डबल आई देला मेटाफेज नाई भूल अच्छी एटा हला प्रोफेज एटा हला मेटाफेज सो करेक्ट ऑप्शन हला बी DNA replication takes place in S phase. If the initial amount of DNA is represented as 4C, then it increases to dash during S phase, that is 8C. Double synthesis है जो तो DNA कथा कुआ जाउची तो से double हवा synthesis हवा. Then 14. If A is the amount of DNA present in a cell after mitosis, the amount of DNA per cell in G phase will be 2A. DNA रा amount double हवा करना synthesis होची. During which phase of cell cycle amount of DNA in a cell remains 4C level if the initial amount is denoted as 2C, that is G2MM. फोर सी लेवल रे रहइबो. In human somatic cells, DNA replication takes place in the nucleus and centriole duplicates in the cytoplasm of the cell. In S phase of the cell cycle, amount of DNA doubles in each cycle. Correct. Which of the following statement is correct? If a cell contains twice as much DNA as in the functional cell, that is option B. Cell is preparing to divide. The centrosome duplicates during the S phase of cell cycle. Statement 1. S phase is called synthesis phase. In S phase, the number of chromosomes in a cell remains the same. So 20 number कौन होगा? ना statement 1 दला S phase is called synthetic phase. ठीक अच्छी. Then दला in S phase, the number of chromosome in a cell remains the same. क्रोमोजोम कथा अच्छा क्रोमोजोम कथा देइछे एटी मार्क करबा आमे आ पूर्वरु आमे कहूचे डीएनए जे कि कोन हिजबो डबल हबो हला 2C रहिथिले से 4C हबो किंतु क्रोमोजोम जेहेतु आमे क्रोमोजोम डिटरमाइन करूचे केमती ना ऑन द बेसिस ऑफ नंबर ऑफ सेंट्रोजोम हला तले एटी रहला गोटिए सेंट्रोजोम ताहले आमे कहबा गोटिए क्रोमोजोम जेतले से सिंथेसाइज करबो ताहले भी आमे एटा को गोटिए क्रोमोजोम बोली कहबा किंतु डबल हिजबो डीएनए सो क्रोमोजोम कथा कहिछि ताले ठीक अछि बोथ स्टेटमेंट 1 and 2 are correct. Then 21 number. A somatic cell that has just completed the S phase of its cell cycle as compared to gamete of the same species has twice the number of chromosome, twice the amount of DNA. Bhul. Same number of chromosome but twice the amount of DNA. Twice the number of chromosome and 4 times the amount of DNA. somatic cell katha deichi, just completed S phase of its cell cycle as compared to gamete of the same species. 4 times of the number of chromosome and twice the amount of DNA. Nahi ତା ଭିତରୁ correct answer ହେଲା C, twice the number of chromosome and 4 times the amount of DNA. During the cell growth, DNA synthesis takes place. S phase. During which phase are spindle proteins synthesized in preparation for mitosis, that is G2 phase. Spindle protein. Out of the following, which is called quiescent phase, that is G0. Cell in G0 phase exit the cell cycle. Cells in the quiescent stage remain metabolically active. Some dividing cells exit the cell cycle and enter vegetative inactive stage. This is called quiescent stage. This process occurs at the end of M phase. Assertion. The G0 phase of the cell cycle is called quiescent stage. Correct answer. Then, in the G0 phase, cells are metabolically inactive and continue to proliferate. तो आम ये कहले G0 रे मेटाबोलिकली एक्टिव रहिबो, तो ये कहला मेटाबोलिकली इनएक्टिव रहिबो, ताले असर्शन भूल, सॉरी रीजन भूल, असर्शन ठीक. So assertion is true but reason is false. C. Assertion author at 28 number. हाँ. Then in certain cases cell enters in G0 phase. G0 phase को cell enter करे before S phase. Cells which are not dividing are likely to be at G0. Active रहे किंतु divide हुए. Mitosis is characterized by equal division. Shortest phase in cell cycle is mitosis. Diploid somatic cells divide by mitosis. When cell has stalled DNA replication fork, which checkpoint should be predominantly active? That is G2M. एटा मन रखिबा. G2 और M. Next, from the following, identify the correct sequence of phases of mitosis. Prophase, telophase, anaphase, metaphase. तो mitosis पई हमरो correct sequence हला prophase, metaphase, anaphase, telophase. Select the correct statement related to mitosis. ओटा आसीबा हमरो option आसीजिबो B. Amount of DNA in the parent cell is first doubled, then distributed into two daughter cells. Karyokinesis is the division of nucleus. Identify the phase that is characterized by the disappearance of nuclear membrane and nucleolus, that is prophase. During prophase, chromatin network condenses. Duplicated centriole move to opposite poles of a dividing animal cell during prophase of the cell cycle. Duplicated centriole move to opposite poles of dividing centriole कथा पचार. Then during cell division the spindle fibers arise from centrioles. Spindle fibers formed during cell division are made up of microtubules. Identify the proteinaceous component of the cytoplasm that helps in the initiation of the assembly of the mitotic spindle, that is microtubules. Spindle fiber attached to kinetochore of the chromosome. During metaphase, chromosomes get arranged at the equator. Metaphase मन रखो middle equator रे arrange हो. The best stage to count the number of chromosomes during mitosis is a structure of chromosomes can be best seen at metaphase. Attachment of spindle fibers to kinetochores of chromosome becomes evident in metaphase. The stage of mitosis in which chromosomes begin to separate and move to the two poles of the dividing cell is called as anaphase. अपोजिट पोल को मूव हम क्रोमोसोम एनाफेज देन ये करेक्ट ऑप्शन चूज करवा G1 फेज G1 फेज रे आसी जीवो सेल ग्रोथ एंड ऑर्गेनेल डुप्लीकेशन एस फेज डीएनए रेप्लिकेशन एंड क्रोमोसोम डुप्लीकेशन ठीक अच्छी G2 फेज साइटोप्लाज्मिक ग्रोथ ठीक अच्छी मेटाफेज इन एम फेज अलाइनमेंट ऑफ क्रोमोसोम सबू पॉइंट गुड़ाक आमर ठीक अच्छी सो ऑप्शन ए सॉरी ऑप्शन सी I A double I B triple I C I B D movement of chromosomes during anaphase is dependent on association of spindle fiber with kinetochores. Correct answer. Chromosomes move to the respective poles due to a contraction of spindle fibers. A stage in cell division is shown in the figure given below. Identify the stage correctly along with its characteristic. अच्छा एनाफेज स्पिंडल फाइबर कंट्रैक्ट डॉटर क्रोमोसोम पुल्ड टुवर्ड्स पोल ठीक अच्छे टेलोफेज क्रोमोसोम अलाइंड एट द इक्वेटर भूलेगला प्रोफेज कॉइलिंग ऑफ डॉटर क्रोमोसोम न्यूक्लियर मेम्ब्रेन रीअपियर नुह डिसअपियर देन मेटाफेज resting phase cell does not undergo any change good so option A chromatids move to opposite pole during anaphase promoting complex APC is a protein degradation machinery necessary for proper mitosis of animal cells if APC is defective in a human cell which of the following is expected to occur chromosomes will not segregate APC जदि डिफेक्टिव हे जिवो ताहले कोन हो ताहले क्रोमोसोम सेग्रीगेट हबो नाही mitotic anaphase differs from metaphase in possessing same number of chromosome half number of chromatids mitosis रे same number of chromosome half number of chromatids which is not true for anaphase of mitosis nucleus reappears not true reappearance of nuclear membrane, disappearance of spindle fiber, formation of chromatic network are the characteristic of telophase. A stage in cell division is shown in the figure. Okay. अच्छा एटी कौन होची हमरा telophase nuclear envelope reform Golgi complex reform so ठीक अछि late anaphase move away equatorial plate Golgi complex not present cytokinesis cell plate formed mitochondria distributed between two daughter cell telophase देइथिला कोन क्वेश्चन हमको select the answer which gives the correct identification of the stage अच्छा से stage रो correct देछि हां so obviously एटा हला हमर telophase फिगर आई एंड डबल आई रिप्रेजेंट कौन करुच्ची ना आई रे देखबा हमें क्रोमाटिड सेपरेशन हेची जेतुटा माइटोसिस क्रोमोसोम नंबर सेम अछि क्रोमाटिड सेपरेशन हेची तो एटा हमर ऑप्शन आसीबो नंबर आई लेट एनाफेज डबल आई प्रोफेज In animal cell, cytokinesis takes place by furrowing of plasma membrane. Animal cell. यदि plant cell इतांदा ताहले हमें कोई तांदे cell plate. Cytokinesis in plant cell takes place by cell plate formation. A cell at telophase stage is observed by a student in a plant brought from the field. He tells his teacher that this cell is not like other cell as telophasic stage. There is no formation of cell plate and thus the cell contains more number of chromosomes as compared to other dividing cell. So this would result in polyploidy. तो ये पॉलीप्लॉइड कहीं बाछा देख नो फॉर्मेशन ऑफ सेल प्लेट फॉर्मेशन हो ना जदि गोटे सेल रे सेल प्लेट फॉर्मेशन हम ना ताले से डिवाइड हेनी ताले से नंबर ऑफ क्रोमोसोम कौन है बेसी होगला ताले से जो डिजीज आसला सेटा हला पॉलीप्लॉइड मोर देन तमरो जेमते आमे कहि परिबा पॉलीप्लॉइड रे 3n फॉर्मेशन होबा 5n फॉर्मेशन होबा 6n फॉर्मेशन होबा दैट इज पॉलीप्लॉइडी कंडीशन जेमती कि आमे एटा कहि परिबा पॉलीप्लॉइडी कंडीशन रे तमरो 47 नंबर ऑफ क्रोमोसोम 48 नंबर ऑफ क्रोमोसोम है डाउन सिंड्रोम क्लेइनफेल्टर सिंड्रोम ये सब आमर पॉलीप्लॉइड रे जाउची देन 29 नंबर syncytium is the result of karyokinesis not followed by cytokinesis ठीक अच्छी in plant cell middle lamella formation start from center grows outwards of the cell leads to cell wall formation ठीक अच्छी किंतु कई कार explanation नुह so option B which of the following options gives the correct sequence of event during mitosis condensation nuclear membrane disassembly then the crossing over segregation telophase condensation nuclear membrane disassembly arrangement at equator centromere division segregation telophase correct it is the crossing over so bolega arrange the following events of mitotic cell cycle in series amount of DNA per cell double number 1 then condensation of chromatin into chromosome number 2 disappearance of Golgi complex and ER number 3 separation of chromatids at center number 4 then reformation of nucleus and Golgi complex number 5 then last one chila number 6 eta distribution of mitochondria and plastic to daughter cell so number double I then IV then triple I then VI then I then V option C given below are events that take place in mitotic telophase arrange them in the correct sequential order and choose the correct option okay correct option हमको choose करार अछि न्यूक्लियोलस गोल्गी कॉम्प्लेक्स एंड एंडोप्लाज्मिक रेटिकुलम रीअपियर कोथरे हमको देखिबा माइटोटिक टेलोफेज रे रीअपियर ठीक अछि देन क्रोमोसोम्स क्लस्टर एट ऑपोजिट पोल नाही न्यूक्लियर एनवेलप assembles around the chromosome cluster ठीक अछि देन एटा देखिया कौन सी क्रोमोसोम क्लस्टर एट द ऑपोजिट पोल ओके ऑपोजिट पोल देछि सो ठीक अछि एटा करेक्ट ऑप्शन जो खोजिबार अछि हां न्यूक्लियर एनवेलप assembles around the chromosome cluster ठीक identify chromosomes is lost as discrete element ठीक अछि सो ऑप्शन बी तो ऑप्शन बी प्रथमे कोन होबो क्रोमोसोम क्लस्टर होबो एटा हला नंबर 1 देन द आइडेंटिफाई क्रोमोसोम इज लॉस्ट एज डिस्क्रीट एलिमेंट एटा नंबर 2 न्यूक्लियर एनवेलप असेंबली होबो नंबर 3 देन एटा रहबो नंबर 4 लास्ट रीअपियर हाउ मेनी mitotic divisions are needed for a single cell to make 128 cell that is 7 mitotic division Then next, identify the number of chromosomes present in a human somatic cell during G1S M phase respectively. Number of chromosomes present in a human somatic cell, that is 46, 46, 46. That is somatic cell. Two daughter cells formed during mitosis contain the same amount of DNA and the same set of chromosomes as those of parent cell. Option B. The process of mitosis can be studied in onion root tip, garlic root tip, tendril tip. So all of the above. DNA replication in bacteria occurs during S phase within a nucleolus prior to fission. Which of the following is not a characteristic feature during mitosis in somatic cells? That is option B, synapsis. क्रोमोसोम मूवमेंट हुए, स्पिंडल फाइबर हम, न्यूक्लियस डिसअपियरेंस हम, बट साइनेप्सिस माइटोसिस पचार. Which of the events listed below is not observed during mitosis? माइटोसिस कथा हमें पढ़ुचे. सो क्रोमाटिन कंडेंसेशन हुए, मूवमेंट ऑफ सेंट्रियोल टू अपोजिट पोल हुए, अपीयरेंस ऑफ क्रोमोसोम विथ टू क्रोमाटिड्स जॉइंट टुगेदर एट द सेंट्रोमियर. ठीक, क्रॉसिंग ओवर हुए नहीं, क्रॉसिंग ओवर मेयोसिस हुए. The next आसिबा, which statement is true about mitosis? After mitosis, the number of chromosome remains same. ठीक, mitosis can take place in haploid cell. नहीं, mitosis can take place in both haploid and diploid cell. ठीक, option D. Old and worn out tissues are replaced by mitosis. The diploid somatic cell divides by mitosis. Main significance of mitosis is that it helps in growth and multiplication in organism. In the meristematic cell of shoot apex, division will take place by mitosis. Cell growth disturbs the ratio between the nucleus and cytoplasm. Cell growth. Then mitosis plays a significant role in cell repair. दुईटा जाक ठीक अच्छी. So ये option हबो B. Both assertion and reason are true but not the correct explanation. Choose the correct alternatives. Mitosis is observed only in the diploid somatic cell. Mitosis is observed in the diploid cell of animal. ठीक है. Then mitosis is observed in both the haploid as well as the diploid. एटा मध्य ठीक अछि plant cell रे. In some social insect, haploid cells also divide by mitosis. एटा भी ठीक अछि. So double I, triple I V are true. Then meiosis. Meiosis occurs in organisms during sexual reproduction. Meiosis is observed in reproductive part. Meiosis in plants and animals lead to the production of gametes with one set of chromosomes. Which of the following phase is longer in meiosis as compared to mitosis, that is prophase? The correct sequence of the substages of prophase 1 is leptotene, zygotene, pachytene, diplotin, diakinesis. Option B. Which is the first stage of prophase 1? Obviously I am कले कौन leptotene. During meiosis 1, in which stage synapsis takes place, that is zygotene. In prophase 1 of meiosis, synaptonemal complex is formed between a pair of homologous chromosomes. केमिति हुए तो ये जो बनिला तो ये दुटा हेले होमोलोगस क्रोमोसोम बट नॉन सिस्टर क्रोमाटिड भितर होउची. देन the complex formed by a pair of synapsed homologous chromosome is called as bivalent. एबे जो complex बनिला ताको आमे कहबा bivalent. Bivalents are formed during zygote. A bivalent of meiosis 1 consists of 4 chromatids and 2 centromere. 4 chromatids and 2 centromere. एट देखिबा 1 2 3 4 ये हला गोटे सेंट्रोमियर ये हला गोटे सेंट्रोमियर. Bivalents in meiosis are tetrad. Tetrad कहीं को वाला करना चारटा रहला 1 2 3 4. In meiosis, crossing over is initiated at pachytene. During which stage crossing over habo Pgt re? Then during which stage of prophase 1 genetic recombination of parental characters takes place? Pgt. Crossing over takes place between which chromatids and in which stage of the cell cycle? Non-sister chromatids of homologous chromosome at Pgt stage of prophase 1. Crossing over. Identify the incorrect statement with respect to meiosis. Incorrect statement. Meiosis involves two sequential cycle of nuclear and cell division, but only a single cycle of DNA replication. ठीक अच्छी. Incorrect statement को जीवार अच्छी. Meiosis involves pairing of homologous chromosome and genetic recombination. ठीक. Between chromatids of non-homologous chromosome. এটা भूल. Chromatid देला non homologous हमको homologous दरकार. This is the incorrect. Then 4 haploid cells are formed at the end of it. ठीक अछि. Then in crossing over, the exchange of genetic material between two homologous chromosomes occurring during both mitotic and meiotic division. Assertion पचारिछि exchange होउछि. So assertion जोटा पचारला कोन कहिला? Two homologous chromosome occurs during both mitotic and meiotic. Mitotic रे हुए नहीं, only meiotic रे हुए. So assertion is wrong. Process of crossing over is only possible and is mediated by recombinase. एटा ठीक अछि. So assertion is false, but reason is true. Option C. What is the the enzyme recombinase is required at which stage of meiosis? That is Pgt. Function of the enzyme recombinase during meiosis. That is alignment of bivalent chromosome on equatorial plane. Then given figure is representation of a certain event at a particular stage of a type of cell division. Which is this stage? एटा हला हमर प्रोफेज 1 ड्यूरिंग meiosis. Points at which crossing over has taken place between homologous chromosomes are called as chiasmata. एमती जो होता है जो पॉइंट आसे एटा हला हमरो chiasmata. Which of the following events takes place during diplotin stage of prophase 1 of meiosis? That is option D, process of repulsion. Coupling and repulsion theory हमें पढ़ंती. This is the process of repulsion. Diplotin stage. The stage during which separation of the paired homologous chromosomes begins is diplotin. Homologous chromosome ro pairing. Dissolution of the synaptonemal complex occurs during diplotin. Which stage of meiosis does the following figures 1 and 2? 1 leptotin, 2 Pgt. Movement of chiasma towards the end of chromosomes is called as terminalization. Terminalization occurs in which stage? Diakinesis. Match the following with respect to meiosis. Zygotin. Zygotin synapsis. Pachytin. Pachytin crossing over. Diplotin chiasmata. Diakinesis terminalization. So option A. Which of the following does not occur in prophase 1? Synapsis, crossing over, chiasma formation, formation of dyad of cell. So formation of dyad of cell. The following figure shows a stage of meiosis. Identify the stage. Metaphase 1. Equator range. During anaphase 1 of meiosis, anaphase 1 रे कौन होगा? Anaphase 1 रे homologous chromosomes separate है. At the end of anaphase 1, each pole of cell has half of the number of chromosome. एठी chromosome number half होगा. Mitosis रे chromatid separation है. The number of chromatids in a chromosomes at anaphase of mitosis and anaphase 1 of meiosis is 1 and 2 respectively. In meiosis, haploid condition is maintained by which stage? Anaphase 1. At which stage of meiosis is the genetic constitution of gametes finally decided? Anaphase 1. In meiosis 1, centromere does not divide at all. Then anaphase 1 recombinase. Anaphase 1 अच्छा अब थोड़ा टी पढ़ी दे कौन है recombinase sorry anaphase 1 sister chromatid associated at the centromere, then chromosome alignment dela, then nucleolus disappear dela, and recombinase dela, splitting of the centromere. Okay, তাহলে আমি অপশন এটি जोड़ीबा diakinesis, nucleus disappear, then Pgt recombinase, then anaphase 2 splitting of the centromere, then asila anaphase 1 sister chromatid associated at the center. Option I, C, B. Option B. The number of chromosomes after first phase of meiotic division, that is become halved. After meiosis 1, the resultant daughter cells have twice the amount of DNA in comparison to haploid gamete. Which of the following is present between meiosis 1 and meiosis 2? Interkinesis. Meiosis 2 is similar to mitosis. The chromosomes align at the equator and the microtubules from opposite poles of the spindle get attached to the kinetochores of sister chromatids are in metaphase 2. In meiosis, the centromere divides during anaphase 2. Arrange the following events of meiosis in correct sequence. Crossing over the synapses, terminalization, disappearance. Sequence हमको करिबार अछि. So प्रथमे synapsis number 1, then आसीबो crossing over, then आसीबो terminalization तो last आसीबो ना. Then कौन सी disappearance of nucleolus भी अच्छी. So option is C. तो आगो आसीबो हमर terminalization, then आसीबो disappearance of nucleolus. Then which one of the following is wrong for meiosis? It leads to formation of sister chromatid. ठीक अछि. It occurs in diploid cell. It occurs in haploid cell. This one is wrong. Occurs by splitting of chromosome, separation of sister chromatids. Then match the changes which occur in the chromosomes during meiosis with the different phases of prophase 1. Pgt recombination nodule, then zygote, pairing of chromosome, then diplotin dissolution, leptotin compaction. So option D. Match of the following stages of meiosis in column 1 to column 2. अच्छा. Then Pgt crossing over, then ଆସିବ zygote ନା sorry metaphase 1 ଆସିବ chromosome align at equator equator plate. So D, एटी दीटा D अछि, C अछि, A अछि. So option B. Triple I हला B, diakinesis, terminalization हबो, then zygote pairing of chromosome. Identify the correct combination of statement. Leptotin में chromosomes become gradually visible. ठीक अछि. Nuclear membrane do not reappear in telophase rule. Anaphase 2 does not begin with simultaneous splitting of the centromere of each chromosome in meiosis rule. So option I and IV. Prophase 1 is characterized by appearance of bivalent chromosome as tetrads in meiosis 1. Then next, match the following. Prophase 1 crossing over occurs लिखिची. Then anaphase 2 sister chromatid separates. Anaphase 1 देला, telophase 2 देला. ताले कोउटा ठीक अछि ना ऑप्शन 1 and 2 ठीक अछि. The equator देछि भूल. Mitosis and meiosis differ from each other because the former can take place in haploid or diploid cell while latter takes place in diploid cell. Meiosis and mitosis differ from each other because in meiosis खाली meiosis रे कौन हुए बोली ना 4 nuclei formed are not similar to parental ones. Homologous chromosome pair and exchange part ठीक अछि. Number of chromosome get up, so all the above. For a particular character in an individual, which gamete gets only one allele? Each gamete gets only one allele. Okay. Then chromatids of a chromosome split and move towards opposite pole during anaphase of mitosis. So it hela both statements are correct, but B is not the reason for A. Then mitosis. Daughter and parent cell have same chromosome and numbers. Column 1 सहित column 2. Meiosis. Both haploid and synapses of homologous chromosome. एटा ठीक अछि. Synapses of homologous chromosomes. So I रो C कले mitosis. एटा double I D. Select the correct match. Zygote synapses. So भूल अछि. Zygote synapses. So only I. When a unicellular zygote forms multicellular organism, which division takes place? Mitotic division takes place. Identify the wrong statement, about meiosis. Wrong statement कोटा अछि. 2 cycles of DNA replication occurs. Which type of cell division occurs in the gonads? Mitosis and meiosis. Then synapses aligns homologous chromosome. The correct option anaphase 2 देला. Then ଆସିଲା synthesis of RNA and zygote. Then ଏଠି ऑप्शन टा नाही लिखि दबा. ऑप्शन एठी हबो कहा संगरे synapses aligns homologous chromosome with zygote stage रे हबो. Then synthesis of RNA and protein G2 phase. Then triple I action of enzyme recombinase Pgt. Then IV do not separate but chromatids move towards opposite pole. Anaphase 1D. Then chromosome numbers is restored by mitosis. Advantage of crossing over is that it causes variation. 4 daughter cells formed after meiosis are genetically different. The significance of meiosis lies in all of the above. Reduction of the diploid number of chromosome to haploid, maintaining constancy in the number of diploid chromosome during sexual reproduction, production of genetic variability in the population of a species. So all of the above. Then meiosis results in production of gametes, reduction in number of chromosomes, introduction of variation, all of the above. Meiosis helps in evolution and improvement of the races. ठीक अछि. Crossing over occurs in meiosis which results in new combination of gene producing genetic variability. Correct explanation. Option A. Which statement best explains the evolutionary advantage of meiosis? Meiosis is necessary for sexual reproduction. Genetic recombination is possible from generation to generation. Correct. Tetik hela amara cell cycle and cell division with plasma membrane structure with cytoskeletal structure and function. So किछि लिमिटेड टॉपिक नहीं थी ता ऊपर एति क्वेश्चन आम डिस्कस कले रिवीजन करनबा एगुड़ाक सब थैंक यू।